Okay. Good morning, good afternoon, good evening. Welcome everyone to this webinar. Thank you so much for being here. My name is Jim Pickett. I'm a senior advisor with AVAC and really excited to welcome you to today's webinar. So let me go through some slides here to sort of set things up. Let me make sure you can see these slides. First of all, hold on. Okay. So if you are at the Global Advocates offer insights and directions on the science and next steps needed for CROI 2022, you are in the right place. So thank you so much. Today we have a number of discussants who will be talking about the science we all heard at CROI. We'll have Harry Tembo from Zambia, Katie Willingham from the United States, Richard Jeffries from the United States, Shobha Shukla from India, and Simon Collins from the United Kingdom. They'll tell you a little bit more about themselves when we get to that part. So thank you all for being here. And Harry, we're trying to get you connected as a panelist. If you want to private message me or email me if you're having issues, but we want to make sure that your thoughtful comments are included in our discussion today. So let me know. We have a little time to figure out any logistics problems we may be having there. So thank you. So I want to first of all thank the groups who created this webinar. It was brought to you by the Community Liaison Subcommittee of CROI, and that includes my colleagues Dawn Everett and Intando Yola. So I know we all can't hear or see each other in this webinar format, but give a shout out to Dawn and Intando. I also want to give big thanks to the European AIDS Treatment Group for helping us pull this webinar together, and to AVAC. And someone mentioned in the chat if it was an April Fool that I was joining AVAC. I did join AVAC on April Fool's Day. So you would think, and it seems very plausible, but it is in fact, it may be an April Fool, but it's still true. All right, so logistics. This webinar is going to be recorded. You consent to this recording by staying here, by attending. We will send everyone who registered today a recording link. Now that's whether you're able to show up in real time or not. If we have your registration, you will automatically get a link to the recording and to the setup slides that Richard Jeffries is going to walk through at the beginning of this call today. We encourage you to chat, and leave questions in the QA. So in the chat, please talk with each other and make commentary. If you want specific questions answered, please put those in the Q&A feature. We're also trying to keep this as casual as possible, even with this large number of folks. So if you want to say something out loud, please raise your hand and we can make it so you can be allowed to speak. So if you want to say something, we'd love to hear your beautiful voice. Just raise your hand. Otherwise, please leave questions in the QA and comments in the chat. We'll try to be keeping track of all of the above, but if you kind of separate what you're doing, comments between comments and questions, we'll at least make sure we can get to the questions. We definitely want to have a very rollicking, freewheeling discussion. So please help us make that happen. Now I just want to give a little promo to something I've got cooking up in my new role at AVAC. I'm going to be a butterfly catcher. Actually, hello, let's make this slide move. Okay. So I'm going to be launching very soon what we're dubbing the Choice Agenda, Hot Topics in HIV Prevention Research. It will be a series of webinars, typically once a month, and a highly active, moderated, robust listserv. If people remember the IRMA listserv, we're going to bring some of that vibe back and create a couple of platforms here between webinars on the list to discover, discuss, and debate all the things that matter to us in HIV prevention research, from the basic science to clinical trial science to implementation science to communities impacted to funding, all the various issues that impact our work, and really with a thought towards a choice agenda, meaning an HIV prevention agenda that creates multiple modalities for all kinds of people, that does not defer to just long-acting and systemic products, but it provides for an array of products that can fit different people's needs at different parts of their life. So, with that all said, folks who have joined these calls before are familiar. Oh, I should just say, stay tuned. We'll be sending you an invite to join the list, and we'll be sending you invites to join the webinars. Please, please, please share those invites widely. This is not an exclusive club. We want to really have tons of folks involved. So any of your colleagues or networks who would be interested, please share invites to listservs and to webinars. We really hope to have create some really robust discussion moving forward about all the really important issues of the day. So please, please, please feel free to do that, and stay tuned. Since you are all on this webinar today, you will be the first to know about all of this stuff. You will definitely be the first. Okay, so finally, the folks who have been on calls with me previously know I like to do a little Zoom Storm icebreaker. And so for today, the Zoom Storm is going to be a fun one. They're always fun, but this is especially fun. For folks who join the call a little bit early, I like to pretend I'm a DJ and have some music to kind of warm us up and get in a good mood. And what I would like to do is crowdsource a choice agenda playlist. So a playlist that you all create, songs that you love, songs that you want to have, you would like to hear on a playlist, songs that we can feature during our upcoming webinars. So with that said, I'm going to ask you to type in. Now, don't hit enter until I say, but please type in a song or an artist that has to be on this playlist, that has to be on this choice agenda, hot topics in HIV prevention playlist. And when I say go, hit enter. All right. So a song or an artist that you want on that playlist, type it in and please hit enter. All right, I'm seeing lots of good responses so far, but there's way more of you than responses, so let's keep it going. I've seen Gaga, Outkast, ABBA, Sexual Healing, which is especially relevant, I think. Keep going, keep going. We want music from every corner of the globe, old music, quote unquote, new music, quote unquote. Please share. And now I'm going to stop sharing my screen. Let's see what's going on here. Thank you for all the feedback. And keep it coming. We have a lot of webinars ahead of us and a lot of playlists to start and end these calls with. So I'm really relying on you for these to be awesome. So please, please, please keep putting that in the chat. Now, without further ado, I am going to go ahead and open up this call officially. Richard Jeffries from TAG has offered to start us off with a general overview, a recap of some of the highlights from CROI 2022. It feels like it happened yesterday. It also feels like it sort of happened a year ago. So let us kind of get back into the CROI mode. And then we're going to be inviting all of our panelists to come on camera. And they'll be sharing the news and the science that was really important to them. And we're going to be talking about why it's important to them and what we can do as advocates to amplify some of the science and some of the research that's coming out, or the lack thereof, what we need to do as a community. So we're going to start off with Richard. Richard, if you want to go ahead and share your screen and make sure you are off mute, and you can go ahead and get started, my friends.
Sure thing. Is everybody able to see the slides okay?
The slides look great and we can hear you beautifully.
Okay, excellent. And obviously this is a big task to try and summarize CROI, so I'm inevitably going to miss things. I think fortunately there's a great group of panelists and even more great people on the call. So please flag anything that gets missed that you think was important. I'm just having a bit of trouble advancing the slides. Okay. So these were the, and the presentation will include quite a lot of links, which I will post in the chat afterwards. So don't worry about catching all those, because they'll be shared after. These were the topics for the Margarita Breakfast Clubs that were hosted by AVAC and the ATG and the Liaison Committee and others during the conference itself. These have a lot more information on these topics and are well worth watching the recordings. I apologize that the slides are just refusing to advance for some reason. Okay. And so to start on one of the major topics from the conference, which was long-acting antiretroviral therapies, there was an excellent plenary by Dr. Chloe Orkin, who also participated in one of the Margarita Breakfast Clubs, talking about Cabenuva, which is the first long-acting antiretroviral combination to be approved. This slide just summarizes some of the developments that have happened with this therapy since monthly dosing was initially approved. So monthly dosing was shown to be non-inferior, so just as good as a standard oral antiretroviral therapy. They later showed also that dosing every 2 months was non-inferior to monthly dosing, but there are a couple of caveats to that I'll get to. Overall, well tolerated. The failure rate, so people experiencing viral load rebound, was around 1 to 1.5%. That looks a bit like 15%, but it's 1.5. Unfortunately, resistance is usually to 2 classes of drugs, so integrase inhibitors and NNRTIs. So that's problematic because it means people have to be switched to protease regimens. Initially, there was an oral lead-in of getting pills for 4 weeks, but there's follow-up studies have shown that doesn't seem to be necessary. There's been no sort of hypersensitivity reactions. And people that enrolled in these trials, because they were interested in injectable therapy, were overall very, very sort of satisfied with it. I think another caveat to that is that obviously these trials were set up to administer monthly injectable dosing, and it may be a little bit different in regular clinic settings. You know, the kinds of experiences that people have in their clinic settings may shape whether they are, you know, how comfortable they're going to be going in every month or every other month for these injections. And so that's something to sort of look at as we begin to see data, which we haven't really seen yet on how these treatments are being rolled out in the real world. And so the big concern is this issue of resistance with to the drugs, which can develop sort of rarely, but does happen. And there does seem to be an increased risk if for people getting every other month. So that's important for people to be aware of. So after 3 years of follow-up, about a 1 in 40 chance of failure and breakthrough versus 1 in 100 for monthly. although there was an association with an unusual HIV variant that's prevalent in Russia, which may have influenced things, but this does require a switch to protease. And so that's something to bear in mind when people are thinking about what kind of dosing regimen might be preferable. Another presentation at CROI about long-acting therapy, but this time in the prevention context, was just cabotegravir alone. There's been two studies published showing it's highly efficacious as prevention, but this was the first study looking at women who became pregnant and stopped receiving the drug, but there was residual drug in the system. There was no sort of big safety flags here, which is reassuring, no congenital abnormalities, and they're now looking at women who carried on treatment through pregnancy. And this slide is from a great presentation at a British HIV Association, a Best of CROI summary, which I also highly recommend. And again, we'll share the link afterwards. The other big long-acting news at CROI was about a Gilead drug called Lena Capovir, which can be given every 6 months. Unfortunately, trials are on hold. There's an issue with the vials that contain the drug, the potential safety concern. But I think there's optimism that that can be resolved. There was 2 trials reported at CROI, Capella, which was in people with a lot of prior treatment experience, and Calibrate, which was people starting for the first time. Good rates of viral load suppression, and CD4 increases, which is quite unusual in people with heavy treatment experience, to sort of get a boost in CD4 cells. The rates of viral suppression in the people starting treatment for the first time in Calibrate were actually mostly over 90%, particularly if people achieve suppression in the first 24 weeks. Discontinuation is relatively unusual and resistance quite rare. And I think a difference with the Cabenuva is that people developing resistance to Lena Capovir were able to switch to integrase inhibitor-based regimens and suppress viral load. An important trial conducted in 3 countries in Africa was the Nadia trial, which was looking at second-line treatment. This has since been published just last week in the Lancet HIV. The important sort of finding from this study was that this idea that people needed to switch to AZT and 3TC as part of a second-line regimen, probably not the case. People were able to do well keeping on tenofovir and 3TC. Some concerns that the 2 regimens were dolutegravir or protease inhibitor darunavir with 2 NRTIs, and maybe resistance was a bit more of a concern with dolutegravir, but this seems likely to cause an adjustment in guidelines which had previously recommended switching to AZT as part of the second-line regimen. Again, this slide is from a presentation at the Beaver review by Dr. Laura Waters, and I recommend that presentation as well. One of the big sort of news stories from the conference, which I'm sure people saw, was about the possibility of another cure case related to a stem cell transplantation. And this was the first time this has been described in a woman, and this was a middle-aged woman of mixed race who developed a life-threatening cancer called acute myelogenous leukemia. It's a cancer of the immune system white blood cells, so the treatment involves wiping out the immune system and replacing it with an immune system from a donor. Stem cells are sort of like the seeds from which the immune system grows. So you have to knock out the existing immune system, which is quite risky, and then give the stem cells to generate a new immune system. And in this case, they used umbilical cord blood, which can be a source of stem cells, which hasn't been used before in these cases. Also, though, with some adult stem cells from a relative to sort of help cover the time it takes for the cord blood units to generate a new immune system. But the good news was that the transplant was successful. After 55 days, all of the new immune system cells had come from a cord blood donor who had this rare mutation which causes resistance of immune cells to HIV infection by most strains, the CCR5 mutation. The cancer is in remission, and ART was interrupted. Antiretroviral therapy was interrupted 3 years after the transplant, and the woman has been followed for over 14 months now with no viral load rebound. So the researchers are being cautious and using the term remission. Some of the headlines, as you saw previously, like in the New York Times, already called it a cure. There's some uncertainty around how long you should wait before defining a cure. For previous cases, it's been about around over 2 years. So maybe some more follow-up to be really certain that this is a case of a cure. But antibodies aren't detectable. The immune system cells resist HIV infection in lab studies, and there's no sort of HIV genetic material detectable anymore. Switching gears to broadly neutralizing antibodies, there's quite a lot of information on BNAbs, as they're called, at the conference. These are rare antibodies that could be identified, which are quite potent in suppressing HIV. There was 2 studies, one by Ollie Sogard and one by Christian Gabler, which reported that receiving 2 BNAbs was associated in small but measurable declines in the persistent HIV reservoir that's considered the big barrier to a cure. There was sort of isolated examples across the 2 studies. There was 3 in total of people who, after a long time after stopping receipt of the BNAbs and stopping antiretroviral therapy, maintained undetectable viral load for extended periods, suggesting that maybe the BNAbs are boosting the immune response and enabling the control of HIV in rare cases. I think the longest follow-up is about 4 years now. Another is 2 years. And these are associated with some measurably improved immune responses to the virus. So that's encouraging. There was a study in infants in Botswana that showed that dual BNAbs could maintain viral load suppression for an extended period in about half of those children. So that may become an alternative approach for treatment in infants, but I think more work's needed to improve the number of people that are able to suppress. And the reason for those, lack of suppression baseline, sort of resistance to these antibodies, so HIV that's already kind of has mutations that prevent the antibodies having an effect, may require people to be tested to see if their virus is sensitive prior to being given the antibodies. There was also one abstract reporting that a BNAb could be given to newborns potentially as a prevention measure against breastfeeding transmission. But again, that issue of resistance may need to be taken into account in terms of like actually testing that approach for efficacy. One potentially important pediatric study presented by Debbie Passot. This is an ongoing study looking at whether it might be possible for infants who are on effective antiretroviral therapy started right after birth, eventually to be able to stop the treatment without experiencing a viral rebound. So sort of HIV remission is the goal, similar to what was reported in the news a few years ago with the Mississippi baby. And this presentation showed that about 1/3 of the infants after 2 years sort of seemed to really have an absence of detectable HIV and negative antibodies and might be candidates to interrupt antiretroviral therapy and see if they experience remission. That stage of the study has been somewhat delayed by the COVID pandemic, but there's a sort of committee that's evaluating whether that might be appropriate for some of these study participants. There was a really great talk at the beginning of the conference by Mark Thompson from the UK about HIV and aging, and I'm not going to read all of the 8 points he lists. I'll leave them up here for a little bit so people can read, and the slides will be shared afterwards. But I think a sort of fantastic list of concerns that are important for people about HIV and aging. And I think the quote at the end of his talk, after decades of surviving, we want to continue to survive, but we also want to thrive, is a really important. And I will share the link for this talk afterwards as well, because I think it's well worth watching. And then a little bit of science, that there was a study from Kaiser Permanente looking at heart attack rates in people with HIV compared to negative people, finding that there seemed to be a bit of a widening gap and slightly increased risk in a more recent period of study over 5 years. So sort of stressing the importance of cardiovascular disease monitoring to avoid the potentials for problems. A study from a cohort in the UK called POPPY of people over 50 finding that a certain class of drugs called anticholinergic, I don't even know how to pronounce it properly, drugs which are used for a variety of things were associated with a greater risk of frailty and recurrent falls. So sort of recommendation that older people should try and avoid use of those drugs. And then sort of more at the cutting edge, there was a poster presentation suggesting that a new class of drugs that are being studied out in HIV negative people as a sort of potentially anti-aging effects called senolytics had sort of positive effects on cellular aging when they looked in laboratory. So cells can get kind of worn out as we get older, and these drugs seem to have a somewhat rejuvenating effect. Jules Levin from NATAP has really been talking about the importance of studying these drugs in people with HIV for some time now, but I don't think studies are underway. So that's sort of a potential advocacy issue to be aware of. And again, I'll post this link, but NATAP, who have really been leading the charge on this, did a CROI update, and there's a recording available. Another study that got a lot of attention, which is potentially very important, is about preventing anal cancer associated with human papillomavirus, called the ANCHOR study. This was a large study that screened a lot of people for precursors of potential precursors of cancer. And when they identified people that had these high-grade squamous interepithelial lesions, they were randomly assigned to receive immediate treatment or monitoring. And the immediate treatment was associated with about more than half, 50% reduction in risk of developing anal cancer. So that may be something that changes practice, and the investigator, Joel Pileski, thinks it should make screening and treatment standard of care for people with HIV over 35. And covering a lot of territory here, the REACH study was a prevention study looking at acceptability of the dapivirine ring and Truvada PrEP in adolescent girls and young women, I think in South Africa. And so people were given one or the other, were given each for 6-month periods, and then for a final 6-month period of the study could decide which one to use. The vast majority were interested in taking up one of the prevention products, and of those, most chose the ring. So that's sort of encouraging for the prospects for the dapivirine ring and other kinds of microbicide-based approaches. And there's a great microbicide trials network explainer. Again, I'll post the link afterwards. Tuberculosis has unfortunately been an example of what can happen when science doesn't really make much progression. And there was some new data about finally some regimens for people with drug-resistant TB that don't have the kinds of severe risks that some of the older treatments have, like hearing loss. And there are shorter regimens. In a study by Médecins Sans Frontières, this newer combination of newer drugs was highly effective. Also a sort of a flag for people needing emergency contraception who are receiving rifampicin for TB treatment need to increase the dose of drug interaction study. And there, in a symposium about TB, there was news about potentially new long-acting drugs are sort of being developed for all kinds of conditions, and TB, thankfully, is among them. And so there's a great symposium presentation on that. And then lastly, just a little bit on COVID-19. Steve Deeks from UCSF gave a presentation at an interactive session on COVID, presenting some evidence that unvaccinated people with HIV may be at a slightly higher risk for long COVID consequences, potentially associated with inflammatory markers. There was a very large study of looking at COVID-19 vaccine boosters, finding that they remained efficacious against severe outcomes in people with immunocompromise. And an Italian study looking at additional doses of mRNA vaccines in people with HIV, and even in people with potentially immune suppression, CD4 counts below 200, there was a boosting effect, although there was a slightly greater risk of maybe not getting so high antibody titers. And I will stop there and go to the discussion.
Great. Thank you so much, Richard. If you want to go ahead and stop sharing your screen, and if we can see our panelists' beautiful faces, that would be great, live and in person. Hello, hello. First of all, I know we all can't see or hear each other in this setting, but can we give Richard a huge round of applause for doing that spectacular overview? Like he said, it's not easy to, quote, do a brief overview of CROI any year, certainly not this year. So many thanks for taking that on and for making it so clear and for sharing so many great links so we can follow up with all of those things. So now I'm gonna open it up. This is how the rest of the time is gonna work. We have almost an hour. So I'm gonna reach out to each panelist and have them share what their highlights were, the things that they were really focused on from CROI. And we'll be discussing some of those items and then, of course, be bringing in your comments and questions from the Q&A and chat. So again, if you have a specific question, put that in the Q&A. If you're making comments, you can leave those in the chat. We also really highly encourage you to raise your hand if you want to speak. We would love to hear other voices here in the mix. So if you want to raise your hand and make a comment, please do that, and I will see you and call you as soon as I can. So to get started, I haven't told anyone how I'm going to start here, but I'm going to go from how my screen is from left to right. And on my farthest left is Shobha Shukla from India. So Shobha, would you like to quickly introduce yourself and tell us some of the things that you really thought were important to take home from CROI this year?
Namaste from India. And thank you, Jim, for making me a part of this fabulous webinar. It is my deep conviction that every humanitarian or health response, whether it be for HIV, tuberculosis, or COVID-19, has to be a feminist response. And it means having a different kind of system that is based on sharing and caring. And I believe that a world of solidarity is a feminist world where everyone, irrespective of their caste, creed, or sexual orientation, can have a dignified and respectful existence. Now, CROI 2022 presented important TB science updates on TB treatment, including the two most historically neglected areas, drug-sensitive TB and latent TB, according to me. That's my perspective. And this TB science presented at CROI is vital because TB, we know, is robbing us of the gains made in advancing HIV treatment science and rollout. TB is the most common opportunistic infection in people living with HIV, and it is a major cause of mortality among them. So I will quickly go through some of, or rather try to unpack for myself and for others, some of the major research updates in the field of TB. And the first that comes to my mind is treatment of drug-susceptible TB, because it's nearly almost after 40 years that we have got something new, different from what we have been calling this gold standard care of treatment. For me, it has never been a gold, it is a standard care of treatment of 6 months. And now the CDC TBTC study 31A5349 has found that a 4-month daily isoniazid, rifampicin, moxifloxacin, and pyrazinamide regimen, that is 2 HPM ZM and then 2 HPM, is non-inferior to the standard 6-month regimen for treating and curing drug-susceptible pulmonary TB in adults and adolescents. Now, treating TB in 4 months, I think that's a big achievement. And in fact, the overall efficacy results for HIV positive subgroups in this study were higher at 85% as compared to 78% in the standard of care arm. This regimen, again, as I said, is the first successful short course treatment regimen for drug-sensitive TB in almost 40 years, and WHO has already endorsed this regimen. Then another study, the SHINE study, which compared 4 versus 6 months treatment in children, both HIV positive and negative, with smear negative non-severe TB of the lungs. And a 4-month treatment of isoniazid, rifampicin, ethambutol, and pyrazinamide was found to be non-inferior to standard 6-month regimen for children with minimal TB. So I think that again is a great achievement. Then for treatment of multi-drug resistant TB, until recently, as we know, the standard of care treatment regimen was 18 to 24 months long and gave a cure rate of 50% with highly toxic drugs and painful daily injections and very, very bad side effects, loss of hearing being one of them. Now, the NICS TB and ZNICS studies have already found an all oral short course 6 months regimen, which we call the BPAL regimen, bedaquiline, pretomanid, and linezolid for highly drug resistant tuberculosis patients. It has found to be safe and efficacious with a treatment success rate of 90%, even in people living with HIV who are infected with MDR-TB. In ZNICS study, the linezolid dosage was reduced from 1200 mg to 600 mg to decrease toxicity, and duration was also reduced from 6 to 2 months to give better results and at least minimize the side effects. So treatment success rate remains the same. Then another study, which has Richard has mentioned earlier, the TB practical study led by MSF, is evaluating a 6 months all oral regimen containing bedaquiline and pretomanid in combination with linezolid, moxifloxacin, and clofazimide. The study design has 4 arms, and the control arm is the WHO standard of care treatment, and then we have a BPAM arm, the BPALC arm, and the BPAL arm. And as of now, the BPAM, that is bedaquiline, pretomanid, linezolid, and moxifloxacin regimen with a tapered dose of linezolid has been found to be non-inferior to this as compared to the standard of care 9 to 24 month long regimen for rifampicin resistant pulmonary TB. And outcomes of the other regimens are expected to be reported soon. Then the ACTG A5375 study determined that a double dose of emergency contraception, levonorgestrel, is required when taken by women who are receiving rifampicin-based treatment for drug-sensitive tuberculosis. So the dose has to be doubled from 1.5 to 3 mg just to avoid the drug-drug interactions. Then TB preventive treatment, which again has been neglected for so long, and that too for MDR-TB. At CROI, we heard about many ongoing studies that are evaluating 6-month MDR-TB preventive therapies for close contacts of MDR-TB patients. The TB JAMP study is examining levofloxacin as MDR-TB preventive therapy in children below 5 years who are close contacts of adult MDR-TB patients. And the Phoenix study is comparing delamanid with isoniazid for preventing MDR-TB in children, adolescents, and adults who are again close contacts of persons with MDR-TB. There is another study, the VQuin study in Vietnam, which is examining the efficacy of levofloxacin for treating latent MDR-TB in all household contacts of patients with rifampicin-resistant tuberculosis. Then at CROI has also shared information on new TB drugs, including long-acting injectables. And wow, what a sigh of relief for many of us, because I think the HIV field has gone much ahead in terms of new treatments. The TB field had been stagnating. We have OPC 167832, which is a DPR-E1 inhibitor, which in combination with bedaquiline and delamanid is being evaluated for safety and efficacy of a 4-month regimen for drug-sensitive tuberculosis. And bedaquiline has been touted as a potential suitable candidate for long-acting injectables. One single dose of long-acting bedaquiline was found to maintain drug and metabolite concentration for up to 3 months in mice, and modeling results expect that this would also be observed in the clinic. Also, next generation diarylpinols like TBA-J876 may be good candidates for long-acting based TB preventive therapy. And long-acting injectable TB drugs, as we all know, will have several potential benefits like improved adherence, greater acceptability, and better integration of TB preventive therapy into other treatment programs, like those for HIV, maternal, and child health. That's about all from me for now. Thank you.
Thank you so much, Shobha, for really a great overview and response to what we are learning about TB. So thank you so much for that. How about we move to Simon? Simon, do you want to give us your high-level takeaways from CROI? And also, before you start, just remind us who you are, where you're coming from, and make sure you are on mute, off mute.
Okay, thanks, Jim. I'm a treatment advocate at a treatment information project in London called iBase. I'll stick a link in the chat afterwards. I've been going to CROI for over 20 years, and one of the things I do is I report from the conference, really important conference. There's usually about 1,000 studies, so we only skim the surface really of the big headline news. And then the highlights are always new drugs or recently approved drugs, you know, as a collection of studies really, and it's important to show that even with really good medicines now, that companies are investing more in making better meds. And so in the US or in high income countries, there's already 10 single pill combinations. And so we still think we can do better. So it's like optimistic for the future. The detail about CROI we'll come back to in the conversation, but definitely we need to talk about the cases of viral rebound with long-acting drugs, because they're just becoming available. And then broader access outside high-income countries where there isn't 10 single fixed-dose combinations. The Anchor study, I think, has headline news about how to about producing a data set that means that for anal cancer we now have the same level of confidence that screening is effective as we have for cervical cancer. And the fact that that data set was missing for so many years, we can come back and talk about that in why the study was so important. And as with everything, CROI will depend on where you live. And so for most people in the world, sometimes where there's not resistance tests or viral load tests, the results from the Nadia study and the VISEN study will change WHO guidelines. The fact there's no advantage in going back on a population level to use AZT, and that recycling tenofovir is good. And there were also, in both those studies also showed that darunavir is clearly a much better first-line protease inhibitor than Kaletra. And so hopefully the guidelines will change there. And also there was a sub-study showing on the importance of additional support. So if your first-line combination has failed, then providing additional support for adherence and other aspects of care increase the success rate of second-line therapy. So all those three things, depending on where you live, will be more practical, of more practical importance. And talking about them in the discussion would be great.
Thank you so much, Simon. I'm going to move to Katie, but before, I'm just going to call out Harry. We see you in the we see you here, and we want to be able to hear from you. So let us know. Try unmuting yourself or raising your hand. We definitely want to hear your words of wisdom from Zambia today. Know you're traveling. Know there may be some connection issues, but please reach out. We see you, but we're not sure what's happening there. So in the meantime, while we're figuring that out, Katie, why don't you go ahead and introduce yourself and tell us some of the things that you were really paying attention to at CROI this year.
Hi, I'm Katie Wineham. I'm with the Well Project. I've blog with the Well Project, and so yeah, I'm coming from a place where you know, I'm not a scientist or a researcher. I am I guess I would consider myself representing the general public. I feel like I'm the public that you want to reach with this information. But I'll admit that a lot of it is over my head. A little difficult to understand, but I can understand most of it. And the things that interest me are a lot of the advancements in COVID-19 treatment strategies that I heard a good bit about, and understanding the impacts of variants on vaccines and the rationale for boosters, because I know a lot of people from my community who I'm going to be writing to are interested in those things. You know, I wrote about the vaccination, the vaccine success rates, and how the vaccines are such an unprecedented success. And you know, we don't yet have the perfect drug, but I did learn that second generation vaccines are in development, and I was really excited about that. And then biomedical advances in HIV prevention, long-acting agents, injectables, implants. That really excites me, because you know, of course, long-acting treatments and cure research are of course big interest to me, being a person, an individual living with HIV. I also got a lot of interest out of the I forget the name of the study, please forgive me, but the children HIV exposed yet unaffected. That was a conversation that really interested me. I guess what I would like to say about that is, I guess, the language of uninfected, infected. If I could say anything about that, I wish we could change the word infected to something else. I think there's a lot of we have a lot of intelligence. I think we could come up with a better term, because this is a term that is offensive to many people living with HIV, because when you think infected, personally, I think of green pus, or you know, turn on the lights and the roaches scatter. You know, I mean, I think of negative things when I hear infection. So I would love if that term could be done away with and changed. I also got a lot of interest in the Anchor study and the REACH study trial. The adolescent girls and young women were given a choice of PrEP for either the ring or Truvada for 6 months, and then they were switched to the other for a while, and then they were given the choice of their preference, and I learned a lot about that, and wrote about these things. I guess that's me for now.
Thank you, Katie. And if I can have the rest of the panel come on video now. we'll kind of open up this discussion a little bit. And again, Harry Tambo, my friend, if you are there, please give us a sign, a symbol. We would love to hear from you. People may remember Harry was a presenter on our pre-CROI webinar and gave some expert tips on attending CROI. He's a veteran and been to many, and gave some very insightful ideas for managing the science and just the overwhelm that CROI can give you. I think to Katie's points, a lot of us can be overwhelmed by CROI. It's a common experience. So Harry, we want to hear from you. If you are there, we kind of see you, but we're not sure if that's a ghost in the machine or what. Anyhow, I want to open this up to the panel. And there's been, you know, and this really resonated in the chat, Katie, your comments about language and using language that can be stigmatizing. And this also brought up, someone earlier mentioned that the language that we use around cure is also tricky, and we have to be really careful with that. There are nuances, and I think too many times we see the word cure used kind of recklessly in headlines, and it leads to not a small amount of misinformation. So I would love to just, who would like to speak to some language issues, whether you may have ideas about what we can say instead of infected or uninfected to be less stigmatizing, and if you have thoughts on how to be better communicators when we use language referring to cure research and what we see are as advances in the cure field. So who would like to jump in on that conversation, on that point? Richard, maybe you haven't spoken since you gave us your overview. Do you have thoughts you'd like to share?
Forgot to unmute. Haven't done that for a while. So I think on the cure topic, it's challenging because I think there can be different issues based on different interventions. So for the people that have received stem cell transplants, I think, you know, there's hope that their new immune system, because it has successfully taken over, is now really inhospitable to HIV. And so there's greater hope that there's not going to be any kind of risk of rebound there. And so, you know, calling somebody cured after 2 years may be, you know, fairly reasonable and justifiable, as happened in the London patient, Adam Castillejo. And I saw a question from Judy Patterson about the BNAB study, where the individual has been suppressed for, I think, 3.5 years, probably longer now. And should that also be called a cure? And I think then it gets fuzzy because there's maybe greater risk of rebound there because the person's immune system isn't resistant. There is some kind of immune mechanism controlling, and there's more uncertainty about whether that might fail at some point, or there might be a sort of an evolution of resistance to the immune responses. So it's really challenging there are different definitions that may apply to different interventions. And I think it would be great if there was maybe some sort of generation of some consensus. Perhaps the IAS, International AIDS Society, would be a good place to do that.
Thank you, Richard. Anyone else want to weigh in on the language issue? I see Simon raising his hand.
So language, definitely. You know, infected should have gone out years ago. We've been, you know, various community advocates have been saying that it's not nice, it doesn't help us, it re-stigmatizes ourselves, and so there's a broader campaign to use first-person language. So we are people living with HIV, or we're people living with hepatitis or diabetes, or all those sorts of things. Much better, definitely. And the cure-related questions, sort of. I mean, understand, but just you stop someone in the street, or you're having a conversation with your mates. You really want to know, you know, is HIV gone? And the geeky, complicated bit is whether there's possibly a cell with HIV in your body or not. We can't test all the cells in your body, so you'll never know that for certain. And scientists who don't like to say prove negatives, prove uncertainties, will always say that maybe it can come back. So it is probably closer to remission than cure. But really, in practical terms, we just want to know, can I stop taking my meds and can I not be infectious to my partners? And there's lots of other approaches that don't require having your immune system completely replaced, which isn't possible. And some of the research into BNAbs is very exciting because they have they work a little bit like HIV drugs and lower your viral load, but there is a big question about whether they also work a little bit like a vaccine. And if they do work like a vaccine, then maybe we could get long-term benefits that run not just for a few years, but that produce a permanent immune response. And that would be really exciting. And the complications from these small case reports where someone uses a BNAb and stays undetectable for a few years, the difficulty is, as long as they're just single people, rather than 5 people out of a group of 10, then we'll never know whether these were natural elite controllers or would have become Visconti patients, which are cases where without treatment, a few people manage to control viral load. And so that's a research question. But all of it's very exciting. Definitely language is really important.
Thank you, thank you. I see Shobha.
Yes, I totally agree with all of you regarding the language issue. And I think, Simon, you have said we have thrown out these terms from our vocabulary and dictionary, but they are very much there. And particularly, I'm sorry to say, in mainstream media, we often find these terms being used unashamedly. I have a lot of problems with other terms also. I have a problem with the term trial, clinical trial, as if somebody is on trial in a court. Why can't we call it a study? It is basically a study and not a trial. Also, terms like, again, they are not perhaps in the they are not in our vocabulary, but still frequently used. Somebody defaulting on treatment. So again, you are putting the person, just putting the person in the dock that you are defaulting on treatment. Even the word like camp, medical camps, camp to me denotes something to do with the army and the military. So there are many other terms, and yes, definitely we need to be very, very careful with the language, which is non-discriminatory. And I think the media needs to be really trained in this, and they need to be careful.
Yeah,
Thank you for that. I just want to. Oh, go ahead, Simon. Go ahead. Okay. I was actually going to bring this up. I see in a comment from David Andrist in the chat, and this is a strangely US thing. I want to see what you all think. The Health Resources Services Administration, which manages the HIV portfolio for the US government, is the big funder, puts out all the money. They're now requiring that we do not say people living with HIV. We have to say people with HIV. And when we have slides, sets, or something that are done under their auspices, they will remove living and just make it people with HIV. And it seems like David feels this way. I personally, hello HHS, hello federal government in the US, I think that's crazy. We actually worked really hard to use living, to have living in our description. And I don't know any people living with HIV. I am one that finds living being a problem, saying with that, or having living in the acronym. I'm not sure where this is coming from, but does anyone have a thought on are we missing something? Is people with HIV somehow clearer or better or less something or more of something else? I really don't understand. Any thoughts? Katie, have you heard this change, or it started really in the last administration, it has just carried over?
I have not heard that. And I don't know. I'm like you. I don't know unless they're just trying to shorten it. But I like living with HIV because living is a positive, and it connotates that we are continuing with our lives. We are living, and living and thriving, and that's a positive connotation that needs to be expanded to the public when people think of people with HIV. They need to think of people living with HIV because it's what we are doing. We are living, we are thriving, and HIV is not what it used to be, and that's still a hard obstacle to overcome because people still have those old ideas of HIV, and the term living with HIV, I think changes a lot of that for some people who who do still think of HIV as a death sentence. Because a lot of people, when they hear HIV, that's the first thing that comes to their mind is death. And so living with HIV works against that. So I like the term as it is myself.
Yeah, I agree. I just, someone mentioned in the chat, I think there's different ways to think about this. Julie mentioned in the chat, maybe this is just bureaucratic shorthand. But, I don't know about you all, but I'm a little tired of bureaucrats making decisions on my behalf without asking folks. Like, I don't know there was no concerted effort by people living with HIV to say, stop saying living, we're just with. Like, that wasn't a thing. If it was a thing, I'm really out of the loop. So stop making decisions but for us without us being involved, right? It's just highly annoying. And yes, Julie, bureaucrats can be tone deaf, and they are tone deaf. And speaking of Julie, I'm going to read a question she had in the chat or in the QA, and then it also relates to some other issues around how we roll this out. Someone had mentioned earlier, just keep this in mind, someone had mentioned earlier about ramping up, viral load testing and availability of viral load testing, and how lack of that could cause problems for some of the rollouts of these new modalities. And that, for one of those, that's long-acting prevention and treatment. So as Julie is saying in the question, we're seeing health providers dragging their feet in the rollout of long-acting treatment in our area. So for context, for people who are not in the US, long-acting treatment Cabenuva was approved last year, at the top of last year, and it's been quite a slow, problematic rollout. It had been approved the year before in Canada, and I don't think they had a great rollout. And then just this year, at the top of the year, the US approved long-acting injectable prevention called Apretude. So Julie goes on to ask, are there activist calls to action that folks are aware of that we can utilize to create more demand and pressure? I truly believe that demand creation on long-acting treatment will help with demand creation for long-acting prevention. Are there any suggestions from the group? It seems like CROI has offered all the evidence needed to rebut any waiting for evidence excuses. So I would love to unpack that a little bit. What do people think we need to do in terms of ramping up demand creation for long-acting treatment and prevention? And then what do we do with recommendations that make it hard to roll out in certain settings? I'll mention that I mentioned the viral load testing issue. So for long-acting prevention, viral load testing is now being recommended as a regular thing to determine whether or not you're HIV positive. So not just a normal HIV test, but viral load testing. And this makes it hard in many settings where viral load testing is not very available. And even in settings where it is, it can make it really challenging. There are a lot of providers in the United States where viral load testing is available who are not happy with this recommendation. So hearing all this feedback, our CDC, the Centers for Disease Control and Prevention, have said, hey, you know, viral load testing is the gold standard, but don't let the lack of viral load testing stop you from prescribing PrEP. So it's a little bit of a mixed message. It sounds like it's hard recommendation, but then there's this sort of waffling around, well, which I'm actually happy for. Don't let this recommendation stop you. But what do people think we need to do in terms of, you know, improving long-acting treatment and prevention rollout? What can we do to create some demand? What are some networks we can engage with? And then what do we think about these, you know, recommendations around viral load testing, et cetera? So who would like to jump in on that conversation? And give me a nod or a wink. I see Shobha.
I just want to make one point, sorry, because we are far removed from what USA and the other countries are doing. I come from India, and I think the first step is for the people and the community to know that these treatments or these options exist. They are there. Unless we are aware of what is available, how can we demand? That is my take on it, coming from India. We don't even self-testing for HIV is not in our national program as yet, and they are still having some small studies, and but it is still not in the program. Although there was some study presented about at CROI only about virtual testing being virtual people through virtual community virtually promoting self-testing and providing self-test kits, and it was a success. But these are small studies, and we still. So I think the first thing is for people to know that these things exist. For us, long-acting injectables and so many treatments, we do not know when they will be available. I was so excited by the dapivirine ring. I do not know when it will come to India. So sorry for this, but what my perception would be very different from the rest of you who are from the Western world.
No, I think that's a really important point, Shobha. That's why we had you here. We want to hear those points. It is not the same by context, and while we all have different contexts, we all need to figure out how to work together and lift all of our boats at the same time, right? So I really appreciate you making those comments. Simon, I think you wanted to say something.
So there's two issues. One is PrEP, one is treatment. Wherever you are in the world, you know, as advocates, we have to be, you know, part of the team that gets information out there so people know what's on the horizon and what's available and what will be available. That's sort of part of our job, you know, wherever we live, really. WHO clearly say that PrEP is a really crucial tool and way to stay HIV negative. You know, and that's the whole world population, really, anybody who's sexually active. And WHO support PrEP as a concept. And the new injectable PrEP clearly is easily as effectable as oral PrEP. Probably, if you have adherence issues with oral PrEP, then it's better. Very few infections happened. The big question about how you get access to injectable PrEP is going to depend on the cost. You know, you've already got activists, Afro-Cab activists, and have called for immediate access to injectable PrEP, not access in 3 or 4 or 5 years' time. You know, the studies that get PrEP approved were run in southern African countries. Now, I've no idea how you price injectable PrEP to be affordable in those countries and also make it available as treatment, because in the US, it's a very expensive treatment, $22,000, I think, something like that a year. And that's not affordable as PrEP, where we've got generic PrEP available. So issues of access will depend on cost and whether generics are involved maybe in producing these injections. But I don't have, as an activist, I can't demand, I can't come up with a solution that will work for a company. So as activists, we have to try and work out what those solutions might be that will expand access. I don't have the answer. I think it's a very difficult problem. And in terms of treatment, I think you're right, Richard. Rollout has so far been slow. It's been slow in Canada, in the US. It's just been approved in Europe. And I think that some of the data that came from CROI is going to be really important in deciding how that uptake goes. Because at the moment you're highlighting 1 in 40 people will have viral load rebound with perfect adherence. And these cases, until we can explain this mechanism, that's a big problem, because some of us stay on the same treatment for a decade. And so, you know, at the moment, the company is working like crazy to try and understand what happens, because it's we've got some background noise.
Yes, we do. I actually think that's Harry. So when you're done with your comments, we're going to see if Harry can come off mute and give us some of his wisdom. But go ahead, Simon. Go ahead and finish.
So the main thing I took from the cases of viral rebound at CROI is that they're not predictable. You know, there's a few caveats. So rollout in the UK has sort of been not put on hold, but there's cautions that say don't run and to use this unless it makes a big difference in your life. If you have real difficulties with pills, then it's a fantastic solution. The benefits will probably outweigh the risks. But this is a combination that's really ideal for someone with adherence problems. And yet, in order to get access to the injectables, you have to be undetectable on oral pills for 6 months. You know, it's a switch option that really we want data in first-line therapy to see if it works like that. And, you know, have to be cautious there. You know, the FDA have been cautious in saying how this could be studied and approved. And if those unexplained viral load rebounds are higher in treatment-naive people, the company might not want to do that research. That would really put access on hold. And then the reason in practical terms why it's complicated is that this was these studies were under perfect conditions. People were retained in study. They came back every 2 months exactly to have their next injections. And 2 months is a short time in monitoring. At the moment, I had my viral load checked once a year. And so it's increased monitoring. And these 2 cases failed, where treatment failed. Their viral load was, one was about 25,000, the other was about 60,000. Now that knocks prevention out of the window. You can't have U equals U if you have a 1 in 40 chance of coming back and finding your viral load is 60,000. Your partner will be positive by then. And I haven't seen many people talking about this. I have seen, you know, with people have focused on the clinical response, that if your treatment fails, you have 2-class resistance. You know, that puts you back to being treated in 2005. You have no integrase inhibitors, and also you have no NNRTIs. And for these, it's only a handful of people it happens in. In the whole studies, it's only maybe a dozen people who have had this sort of viral failure. And so it's a low risk, but if it's you, the implications are really serious. You know, you've got resistance, your partner might be positive. So as an activist or an advocate, I could see guidelines saying, you go onto injectable art, you can't rely on U equals U. And most people now like U equals U. They like to know their partners are protected. They don't have to disclose because there's 0 risk. And so I think this is probably a bigger problem than probably even the resistance.
Thank you for
so
highlighting
huge news,
all those
really.
things.
Huge news and not really covered very much.
Thank you so much for putting a spotlight on that stuff, Simon. Now, Harry, we heard you coming live there. We're in our last minutes, but I just wanted to hear any viewpoints that you may have from your viewpoints in Zambia. Harry, are you able to unmute and share with us some of your thoughts?
Hello, everyone.
Hi, Harry.
And Hi, and apologies for joining late. I'm in Lesotho, and our connection is not very, very good. So I've been struggling to get through. But however, I'm glad that I managed. My input is that one of the presentations I found interesting and which concerned me was about pediatrics, on pediatrics, children who are infected, but themselves are not infected. What was presented there is that research shows that when comparing to children who are born from uninfected mothers and also the infected, that still more the children from uninfected mothers and the uninfected, they succumb to a number of problems and other complications. So what it shows that there's still much to be done, work to be done to improve the quality of lives of the infected mothers. This problem is more pronounced here in Africa because of the poor services we have. So I think it is very important that services in our health institutions and that the systems in developing countries to support the mothers. And to this effect, it means that more research need to be done on how the children, despite being uninfected, but they continue to succumb to a lot of other complications. So we need to advocate that, yes, we are glad that children are born uninfected.
Harry, are you there? Okay, I think we lost you, Harry, but thank you for highlighting those things, and I'm really glad we got to include your voice, even if it is coming up towards the end of our webinar. I mean, I think just
yeah, thank you.
Absolutely, Harry. I'll reach out to you via email. And thank you so much. And I know it's hard to connect while traveling, and I appreciate you really working hard to do that today. I appreciate all the panelists being here today and all of our participants as well. I think just to put to note that, how are we going to roll out? Let's just take long-acting prevention. Right now, which is Apretude in the United States, or long-acting cabotegravir. It is priced at $22,000 per year in the United States. And what's really galling about this price is it is incredibly way too high, right? But it also seems to forget that the United States taxpayer paid for a lot of the research and development of this product. Certainly ViiV kicked in and they paid some money, but the US taxpayer funded some really big studies, hundreds of millions of dollars to develop long-acting cabotegravir as a HIV prevention modality. And yet the United States is being hit with this insane price. It's as high as brand name Descovy. And we're a country that just got access to generic Truvada. So you compare $22,000 a year for Apretude, and you compare that to generic Truvada, which is in our country about $360 per year. It's a huge difference. And can you guess what insurance companies will want to cover? Now, of course, I can't blame insurance companies for wanting to cover the cheaper thing, especially when the cheaper thing works really, really well. But we have to give people options, right? And we have to give people choices. And not everyone wants to take a pill a day. To points made earlier about treatment, it doesn't. If you're not into taking pills all the time, forcing people on the pills is a ridiculous thing to do. So I think we need to be holding pharma more accountable than ever for their pricing. Their pricing of these drugs bankrupts the rest of our systems. If we had more money to put into our systems that weren't being spent on the actual drug and were being spent on service innovations, holistic care, bringing different things together to make accessing care easier for folks, we would be way ahead of the game, but we end up paying our pharmaceutical paymasters far too much money. That's my little rant. We are in our last minutes. If I would like to give everyone 30 seconds to say like the one thing we all should be paying attention to. If you could say one thing, you're in an elevator, it's going down, you have time to say one thing for 30 seconds. What should we all be paying attention to going forward? And this will also help me think about this webinar series I'm planning. I will mine your ideas and be thinking about developing topics and discussions to amplify. So I'm going to start backwards from the way we began, and I'm going to go with Katie, your 30-second recommendation.
A 30-second recommendation is make slides easy to understand. I apologize for the dogs going off right now, so I will go back to mute.
That's a great point, Katie. And we actually heard that the CROI Community Liaison Subcommittee, and we are going to be bringing that forward to the CROI Planning Committee and asking that there be a requirement for presenters to at least have summary slides that help things make sense for people. And so it makes it clearer and easier and so more accessible to all kinds of folks sitting in that room or virtual room. Can we now go to Richard?
Sure. I think one thing is that we would like CROI to be more consistent in enforcing guidelines around reporting participant demographics in studies. And we may be able to share some follow-up action on that, sort of writing to them about particularly race, ethnicity, and sex and gender, because some studies are not really reporting that information at all.
Thank you for spotlighting that as well. That's also something I think we're very keen to take to CROI, and I think it's a bigger issue than just CROI, right? I mean, it's we have poor diversity in so many of our studies, and we've had little to no trans male inclusion in studies. In the US, we have we have disparities around racial inclusion. We have problems around inclusion of cisgender women and transgender women in studies. So the more transparency on that, the better, and we have to push everyone to do better. So thank you for saying that. Simon, how about you?
Talk about what you've heard today. Lots of the best information comes from talking with your friends and tracking the research, and be optimistic that treatments will get better, but we might have to work in order to make access much easier, to get the prices we want, and to make sure that our quality of life is better. And until new options come, be good with adherence, take your meds. If your combination's causing problems, talk to your doctor and ask what else you can use. There's choices in every situation.
Thank you, Simon. And Shobha, I'm giving you the last word. Oh, and you are muted. We have to hear your last words.
Sorry.
That's okay.
Unpack the science for all of us who are not scientists. And please transfer some of the activism to the TB field also. I think when I compare the two, a lot has been achieved in the field of HIV just because of community participation and far greater activism than what is happening in TB. And may our right-wing governments please follow the science. I'm not talking only in terms of HIV and tuberculosis. COVID-19 has shown that science took a backseat in many countries, and we had our own treatments and whatnot. So I think it is very important for the policymakers to follow the science. That's it.
And you know, Shobha, that couldn't be a better wrap up. The overall umbrella, no matter what we're talking about, whether it's health science or environmental science, climate science, is follow it. Listen to the science. And when the science changes, it wasn't because someone lied, it's because we've learned more. Science is a process. Unfortunately, science has been more politicized than ever the last few years. And I think we all have to deal with that. And that might be a really great topic for another webinar. How do we deal with addressing science in this very sort of science-free time where science is just as good as someone else's random opinion? So, hey, I want to thank you all so much for being here. Thank you, Richard, Shobha, Simon, and Katie, and Harry for hanging in there and making sure your voice was heard. Richard, thanks again for doing a really lovely overview. We will be sharing your slides with folks and really appreciate you doing that and also putting together so many links. Thanks to all of you for sharing links. We'll be sending out a follow-up email soon. And in that follow-up email, you'll also be invited. You'll get all these links, and you'll also be invited to join this new sexy listserv that I'm starting. So I hope you will all be there and help make it sexy with me. In the meantime, good day, good evening, good morrow. Take care of yourselves. Keep up the great work, and we will see you soon. Bye for now.
Bye.
Bye. Thank you.
Thank you.
Okay, you good.